Ractigen Therapeutics is moving from a Phase 1 to Phase 2 trial for their investigational treatment for SOD1 ALS. The new study is a randomized, double-blind, placebo-controlled trial using a multiple ascending dose design, a structure that allows researchers to test repeated dosing while carefully increasing the amount of drug given to different groups. This shift from the single ascending dose format of Phase 1 to repeated dosing reflects confidence that the therapy was well tolerated the first time around and that the safety profile is strong enough to justify ongoing exposure. Ractigen's AAN 2025 press release described the first in-human investigator trial in 6 SOD1 ALS patients showed, quote, "RAG-17 was generally well tolerated with intrathecal administration. No dose-limiting toxicities, DLTs, or serious adverse events, SAEs, were reported. Most adverse events were mild and transient," unquote. RAG-17 itself is designed to silence the SOD1 gene using siRNA, essentially acting as a molecular switch that turns off production of the toxic mutant protein responsible for damaging motor neurons in SOD1 ALS. Early findings suggest that Ractigen's SCAD delivery platform maintained drug exposure in the central nervous system after intrathecal administration, which is an injection into the spinal canal. This being a key requirement for any therapy targeting neurons deep within the spinal cord and brain. One of the most closely watched aspects of the early RAG-17 data involves biomarkers, especially neurofilament light chain. This protein is released when neurons are injured, and its levels in blood or cerebrospinal fluid often reflect how quickly ALS is progressing. The fluid that bathes the brain and spinal cord gives researchers a direct readout of what's happening inside the nervous system. Early signals showing favorable movement in this marker with reduced levels in the blood suggest that RAG-17 may be influencing the underlying biology of the disease rather than simply addressing symptoms. Additional early data reported reductions in SOD1 protein levels in cerebrospinal fluid and encouraging shifts in neurofilament light chain. RAG-17 has already received orphan drug designation from the US FDA and has been selected for China's CARE program, reflecting the need for ALS treatments and new therapeutic approaches. Ractigen's ambitions extend beyond ALS. While RAG-17 uses siRNA to silence harmful genes, the company is also developing RNA-based tools that can activate beneficial genes. The same delivery platform that brings RAG-17 into the central nervous system could eventually support a wide range of programmable RNA medicines.